it gives me great pleasure to introduce the next speaker, Dr. Li-Xian. Li-Xian is senior lecturer in the Department of Pediatrics, where her research interests from the time of her PhD studies has been on P53. As many of you know, TP53 is the guardian of the genome, and Dr. Li-Xian is particularly interested in the heterogeneity in the genotype-phenotype correlation between P53 perturbations and the clinical manifestation. Li-Xian is also an accomplished pianist, but we won't get a chance to listen to any of her work. So Li-Xian, let's talk about P53 and the interesting aspects of the genotype-phenotype correlation. Thank you. Go ahead.
Hi, sorry. Okay, thank you very much, Prof. Hani, for your kind introductions, as well as organizers for this platform. Can you all see my slides? Just checking in a full screen mode.
Yep. Yes,
okay.
can.
All right, great. All right, okay, so without further ado, very good morning to everyone. I'm Li-Xian, so I'm actually the scientist from the Department of Pediatrics. So today I'm excited to having this opportunity to share some of the insights about Li-Fraumeni syndrome, which is a very rare cancer predisposition syndromes, which is also in line with today's Rare Disease Awareness Day in PPUM. Okay, so moving on. So Li-Fraumeni syndrome is also called LFS in short, is a cancer predisposition syndrome that is firstly defined back in the 1960s by Dr. Frederick Li, which is the left from the photos, and Dr. Joseph Fraumeni, and hence the name Li-Fraumeni syndromes. So back in those days, both these descriptions were actually based on the observations of families with very specific and extensive cancer histories, specifically looking into bone and soft tissue sarcomas at early age. So unlike other cancer predisposition syndromes, which usually predominantly involve certain organs like breast, like ovaries or colon, LFS is a very unique syndrome whereby it predisposes individuals to a wide range of cancer types, from the head to toe, from the inner to outer. And even some cancers are specific, or I would say commonly associated with Li-Fraumeni syndromes. So such as the adenocortical carcinomas, or what we call ACC, or the rhabdosarcomas of the embryonal aplastic subtypes, or even sarcomas and breast tumors that is presented at a very young age. So back then, Li and Fraumeni actually described LFS using this classic LFS criteria, whereby a person or proband with a sarcomas diagnosed before the age of 45 and having a first-degree relatives like a parents or siblings with any cancers before the age of 45, as well as having a first or a second-degree relatives with any cancer diagnosed at the early age or sarcomas at any age. So, okay, moving on. So here I'm just showing a pedigree of a very classic LFS family. So here, let's say we take the individuals with RMS as a proband. So it was presented with sarcomas at a very young age. And here you can see that he actually have a number of first-degree relatives like siblings or parents, as well as second-degree relatives presented with cancers at a very young age. So this is a very classic examples of LFS families. So only until the 1990s that David Malkin and his team actually discovered that germline mutations of TP53 genes was actually the cause in these LFS families. So like just now, Prof. Hani mentioned, TP53 genes is actually the guardian of genomes. So it's actually the genes that code for the protein P53. So in actual, TP53 is the name for the gene and P53 is the name for the protein. But a lot of time people use it interchangeably. So in humans, we have two copies of TP53 genes, one from the father and one from the mother. So like I say, the protein that's coded by this gene is known as guardian of genomes. So here we can, I know the figures is kind of complex, but what I want to highlight here is that P53 as a single protein, it could actually respond to a wide range of stresses that is shown in the red box, the pink box, wide range of stresses, and it will respond by activating a series of, or a spectrums of cellular processes that is shown in a blue box. Like, for example, it can be repairing damaged DNA, or it can stop a cell cycle arrest, stops the proliferations, or it can bring damaged cells to suicidal apoptosis. So with the ultimate aims to stop proliferations of damaged or potentially cancerous cells. Yeah. So as we know, there are many tumor suppressors genes or tumor suppressor proteins in our body, in our cells. But p53, I would say, is one of the very important tumor suppressors. As you can see over here, it actually interferes with almost all of the aspects of cancer hallmarks. Okay. So now that we know that the LFS, germline p53 mutations is actually what the cause for LFS. So back in the olden days, the genetic testings of germline TP53 is offered to individuals under two circumstances. One is if their family members already tested as positive for p53 mutations, or they fulfill what we call the Schomburg criteria. So due to the limit of time, I won't go into detail what is the criteria, but basically it is a criteria that was established when the researchers realized that the classic LFS criteria that we mentioned just now in the first slides was too strict, and it actually might be miscapture some of the individuals that may carry a germline p53 mutations, and hence they modify it and revise it to a Schomburg criteria. So that was back in the olden days, whereby the p53 gene testing is done at a single genes platform. But as we know that technology has progressed, and now multi-gene cancer testing is multi-genes panel testing is so common right now. So, like, for example, patients that are presented with breast cancers with some family histories, previously, instead of solely testing for genes like BRCA1, BRCA2, now they might be highly likely that they will actually go for a multi-gene panel testing, which often includes TP53, because knowing the importance of it as a tumor suppressor. And even in like commercial testings in the general populations, a lot of time they were looking into risk for heritable cancers and so on. TP53 screenings has often been included. So this progress in technology as well as its applications has revealed a lot more information about germline TP53 status amongst individuals. And because of this, now that we know that the prevalence of this germline p53 mutations is actually could be up to 10 times higher than what we previously estimated from the clinical based kind of family studies. So then that leads us to questions whereby, does presence or a carrying of a germline p53 mutation certainly indicate or certainly predispose these individuals to a cancer development at young age? So in 2017, if I'm not mistaken, the study done by Chee and his teams, they're looking into the a complete TP53 germline datasets by the IARC. And here they show that the penetrance of these germline mutations is actually not fully complete. So you can see the figures on the left. So here it shows that the y-axis is the cancer risk of individuals, and the x-axis is across the age of years. So here we can see that by age of 80s, the cancer risk will be about 80%, which, or penetrance, we call it penetrance, is about 80%, which means that it could be 20% of this germline mutations carrier may not even develop any cancers throughout their lifetime. So of course, here we also shows that the discrepancy or the penetrance might be affected by factors like genders, by age, and so on. And on the right, I would also like to bring to your attention is that, first thing, the germline p53 mutations or variants can be present in many different forms. There are many types. It can be missense mutations, meaning that it leads to a change in amino acids. It can be at different positions of amino acid, or it can be a null or a stop codon. or it can be a frame shift mutations that eventually leads to a failure of production of protein instead of a production of abnormal proteins. So, but here we can see that here they were just showing, I think, top 14 hotspots of mutation that is found in germline carriers, whereby they look into penetrance at 30 years, 50 years, 70 years. So let's say we're just looking into 30 years penetrance. You can see that some of the variants, for example, the one at row 2 and row 4, R273H and R248W, which are the hotspot mutations, they confer a 56 to 58% penetrance. Whereas the variants that are shown as the last row, R213Q, it actually only have a 21% penetrance rate, which is about half of the others variants. So here we shows that even though all these variants, they have been categorized as pathogenics or lightly pathogenics, but these different variants will still express different clinical phenotypes at a different age groups. It can vary from asymptomatic to a striking family history of multiple cancers among the members. So just now we have looked into different TP53 variants may confer a different cancer risk in individuals. Then the next question will be, how about within a family, the member of families which carry the same TP53 mutations, would they share the same level of penetrance? Would they share the same cancer risk? So here I would like to show you, like to discuss with you all if one of the case that one of the LFS kindred that was presented to our centers years ago. So here we can see that here we're just showing a pedigree of 3 generations. So looking at the generation 3, the proband, individuals 1. So this proband was actually presented with embryonal rhabdomyosarcoma at the age of 8 months. And she has a sister with adenocortical carcinoma ACC, which is a classical LFS cancers. And looking at their family history, their mother and as well as their grandmother actually have a breast cancer at their 30s. And their aunt was actually found to have osteosarcoma at the age of 26 years. So here we can see that again, these families fulfill the criteria of a classic LFS. So then upon testing of the germline TP53 testing, we found out that these individuals with presentations of cancers actually carries 6 base pair insertion mutations in the TP53 genes that causing them to have insertions of additional 2 amino acids. So likely that they'll be producing abnormal P53 proteins. So then later on after we also screen their family members. And here we can see that there are individuals who were not presented with cancers, but carry the same mutations. So these are the individuals denoted with a dot. So you can see the individual 5, 6, 7, 8 in the generation 3, as well as their uncle in the generation 2. Individuals labeled as 4. So here is an example of so these individuals has remained cancer-free. So this is an example of a discordance cancer phenotypes that we see in a family, despite them sharing the same P53 mutations. So we were in the effort of understanding what could be the factors that contribute to these variabilities. So first, we conducted whole genome sequencing on these individuals, mainly all the individuals in the generation 3, as well as their parents, individual 1 and 2 in generation 2, as well as the uncles, the aunt who passed away due to osteosarcoma, as well as the individual 8 in generation 2. So first, we tried to figure out whether few of the previously known modifiers or factors that could contribute to this variable penetrance, do they apply to these families? So like we know that these individuals with the mutations, they actually inherited the TP53 mutated copies from the mother. So one of the hypotheses will be that would the copies, the P53 copies inherited from the father that contributes to the differences. So using the WGS, we found out that the individuals 1 in generation 2, individuals 1 and individual 5 actually inherited the same haplotype from the father, whereas the individual 4 and 6 inherited another haplotype. So here we could see that paternal P53 haplotype is unlikely to explain the difference in cancer status between these individuals, among these siblings. Then moving on, we also try to explore some of the polymorphisms. So these are the polymorphisms that has been previously reported to be associated with early onset of cancers. So one of it is actually a polymorphisms in the P53 intron regions. So as you think, usually intron doesn't, variations or changes in introns wouldn't have big impact. But in this case, studies have shown that these polymorphisms in the intron tree actually affects the gene splicing and hence may have an effect on early cancer onset. And the other one will be actually a polymorphisms in the promoter of the MDM2 protein. So MDM2 is actually upstream regulators of P53 that actually tends to degrade P53. So to ensure that the activity actually stops when it's not needed. So we try to explore if these two polymorphisms could contribute to the variable penetrance in this family. But again, none of these polymorphisms were actually detected in the families. So moving on, we also look into whether expanded copy number variations would be the factors, but it shows that from the WGS data, the copy number variations in all the individuals in the same generations or even between generations are fairly similar. And we also conduct a Southern blot to look into whether telomere shortening could be the potential reasons contributing to these differences. But again, we see that there is no significant difference in terms of the telomere length among individuals with cancers and individuals without cancers. So then we then look into rare variants, and we found out that there was a list about 11 rare variants that were actually inherited from the father, the non-carrier fathers, that were inherited by the individuals, by the children with cancers, but not inherited by the children without cancers. So of course, this list of variants still requires more validations studies. But then at least this shared some lights or some ideas as to these rare variants may act as what we call the modifier genes or genetic modifiers that could potentially influence the age of cancer onsets in germline P53 carriers. In this case, the children of these families. Okay, so meaning just now we have seen that different P53 variants may present with different cancer phenotypes or cancer risks. And from the family 1, we found out that even with the same P53 variants, but other variants in other genes may also modify this penetrance of these P53 mutations. So here I'd like to present another family whereby this is actually a case involving a monozygotic twins. So here we're looking into the proband, which is the individual 10 in generation 3. So what happened is that this proband was actually presented to our center with rhabdomyosarcomas at the age of 3 years. And 12 years later, she was presented with osteosarcoma of her right knee, diagnosed at 15 years. So upon checking for her family history, we found out that her eldest brother actually died from a rhabdomyosarcoma at a very young age of 3. And the father, as well as the paternal grandfather, actually succumbed to gastric cancers during their 40s. So Fortunately, she survived through from her osteosarcomas, but at her 21st years, she actually developed a non-metastatic osteosarcoma of the vertebrae. And that time, the prognosis by the oncologists were poor, but fortunately, she survived through. But 5 years later, she developed an adenocarcinoma of colon, whereby she undergone a colonsectomy followed by chemotherapies. And 3 years later, she actually developed her fifth cancer, which is a low-grade oral myofibrosarcomas at the 29 years old. And unfortunately, she did not survive through this, and she passed away at the age of 30, which is actually last year. So while she was presented with 5 cancers, her monozygotic twins, the individual's 9, has remained cancer-free throughout until today, despite both of them being raised up in the same environment. So we tried to perform the WGS on these twins together with the mother, and we have confirmed the status of their monozygotic twins with no significant difference in terms of their genetic compositions. So eliminating the possibility of genetic modifiers in these monozygotic twins. And we also didn't find any differences in terms of their CMV copy number variations. And we could also rule out the possibility of mosaicism between these monozygotic twins, because with the presence of a very strong family history, likely suggesting that the p53 mutations is actually inherited from the father or even from the grandfather. So to explore further outside of the scope of genetics, we actually delve into the epigenetics area. So we examine the global methylation status of their blood tissues. So although we see some of the patterns or different methylation patterns between these two individuals, but as for now, our current findings are still very inconclusive. And our next plan or next move is to actually enhance our study statistical powers by combining the analysis with other Li-Fraumeni syndromes from the database with families that showcase a discordance phenotypes as well, although they are not monozygotic twins. So collectively, from what we understand from these two families as well as findings from the databases, it suggests that there is a complex interplay of genetics as well as epigenetics or non-genetics factors that strongly interact with these p53 mutations. So it creates a biological context that either allow or we say permissive or restrictive for the presentations of cancers in Li-Fraumeni syndromes. So, okay, to sum it up, so as we mentioned, the genotype and the phenotype links in LFS is actually not straightforward at all. So we have from all these different family experience, this we have found out that factors like, for example, like we say, the different p53 variants or age or genders or genetic modifiers or non-genetic modifiers actually plays a role, could be playing a role leading to a differential cancer risk in these germline mutations carriers. And these incomplete penetrance make it very challenging for clinicians to manage these LFS individuals because it's very difficult for them to actually predict the age of onset or the type of cancers that these individuals might be developing. And it makes the whole surveillance of these individuals to be very extensive and also, of course, very costly. And due to all these uncertainties, there's also a lot of ethical issues regarding genetic testing of the minors or challenges in finding the most effective or effective treatment strategies. But of course, our understandings of this LFS is continue growing as we've seen throughout and is evolving. There's a lot more that we can explore. Like first, a lot of efforts has been looking into refining the categorizations or the interpretations of p53 variants, like to see whether how can we better precisely assess their cancer risk in these LFS individuals so that we don't over surveillance or under surveillance them. Or by understanding modifiers or markers, biomarkers, is that possible for us to predict what is the age of onset or the site of the cancer progressions later on in these individuals? So with that, I would like to say that the journeys of these LFS individuals or families are actually very challenging. They are facing uncertainties as well as confronting like shadows of cancers constantly, every day. And that actually emphasizes an urgent needs for us, the scientists, the oncologists, or the genetic counselors, the primary care clinicians, the nurses, or even the patient support groups to come hand in hand with patients as well as the families to try to pave the way for us to refine either the surveillance protocols as well as better understand LFS so that we can come out with a more effective treatments. So before I end, I would just like to take these opportunities to thank all our LFS patients, families for their support, their generous participations in all our research studies, always generally sharing their biological samples, their information for us to better understand this disease and moving forward. All right, if that thing, I will end my sessions. Thank you. Happy
Thank
to
you,
take
Liz.
any questions.
Okay, thank you, Lisian, for keeping to time. We have like 1 minute for questions, but I think you have summarized it very well at the end that the journey to understand this genetic genotype and phenotype correlation is still very crude, um, and especially illustrated by that pair of monozygotic twins. Can I invite any questions from the virtual audience? Okay, if not, then can I just invite everybody to come to the lobby of KWKK because there is a rare disease exposition, and it's in conjunction with rare disease day. So I think you can catch some of the newer therapies as well as the genetic counseling, as well as the genetic team down there. Okay, with that, thank you very much, everybody, and have a good day.
Thank you. Thank you.